Hey guys, welcome back to a new video. In the last video, we did discuss about the classification of virus on basis of genome. We just looked into an overview and different sort of groups including in it. And today's video looking into the DNA. This thing, discussing about this thing, the DNA. First of all, uh, as we know that, uh, you can understand this thing that the viruses which do have DNA as a genomic material are termed as DNA viruses. We have got for the two categories. I'm making a section over here to cut the both screens and not mix that up and don't get confused. So uh, what we have got for the two categories. Let me draw for the two categories. So uh, we've got like we've got the double stranded. I'm just writing DS to understand that double stranded DNA virus, and we also got on the other hand here we've got single stranded DNA virus. So what happens with the double stranded? Mm, what happens is uh, they have got double stranded DNA is general material. So do remember that whatever the virus is, its first priority is to synthesize mRNA protein, and when it synthesizes mRNA, it would be able to replicate. So this, when the virus enters into a host cell, the first target is to uh, you know uh, synthesize mRNA. So uh, even though there are cases when virus is unable to do that, so we're looking into that. But in DNA, double-stranded DNA virus, uh, first step when it enters the cell, uh, synthesize synthesize uh, mRNA using host cell machinery. This is kind of a thing, a host cell. So probably the host cell is also double-stranded. So this double-stranded DNA virus founds a perfect fit, and that simply uses the host cell machinery. It does not need to synthesize anything. The host is double stranded, it is also double stranded. It just uses the machinery of the host cell, and it just makes mRNA in order to replicate and increase its survival chances. So it's host cell and the enzymes present in the host cell. Furthermore, we have got some of them. uh, you know this is quite of a simple thing. uh, it is seen that some of some of them even cause virus, cause cancer. This is something which should not happen, but that is happening. Like so, it is seen that they are also causing cancer in organisms. Don't infect plants. Do remember that double-stranded DNA virus don't infect plants. It is not known that any double-stranded DNA virus has infected a plant. Don't infect plants. It may happen in future. We do get to discover this thing. So what examples? The examples include smallpox and herbs. Right. So I do hope that this side is clear. Uh, or from this side, this is uh, single-stranded DNA as a genomic material. Uh, as I said, that first priority of a virus is to synthesize mRNA, but the single-stranded DNA virus is unable to do that because there is a sequence difference. Like one is single-stranded and another is double-stranded. They are not matching. So first of all, what is as it enters the host cell, In order to synthesize mRNA, they convert into double stranded after uh, entering the host cell. In order to synthesize mRNA, right? So uh, they became double stranded then, and as they do that, they uh, they they synthesize mRNA then. And why does that? As I mentioned before, to continue its survival. So the mRNA synthesis takes place, and this happens the same. Uh, the use host cell all mechanism to convert from single stranded to double stranded DNA. So we have also got a point over here that these are sort of biases that use host cell. Okay, so we need to consider this point because uh, we've got another point of examples. So this first of all, they do convert into a double stranded, and they do have that ability, and they have got the exams, and the examples of it include uh, we can say the parvovirus. Yes, that was parvovirus. And we have got the mild rash virus. So uh, I do hope that you've learned the classification. We've got two types in further videos looking into mRNA, the RNA classification, that is having further many types. And we do hope to meet in the next video. Till that, goodbye.